Right, can you guys see the slides? Okay, so yeah, thank you for invitation. It should be fun to talk to people that may not share your background. I'm a physicist, so the terms that I might say may reflect that. Nevertheless, I'll try to deliver the ideas in the way that biologists and machine learning scientists can understand. Now, the main point of the talk is to talk about only one paper, which discusses the thermodynamic interpretation of evolution and the origin of life. Yet, I will have to introduce a little bit before that, hopefully quickly, is how the learning concept actually falls into that paradigm. And so that's why this is the title of the talk. Now, I'm currently in transition from the NIH to ANC. And so if you are not able to catch me in NIH, it means I already left. If you are not yet see me in ANC, that means I'm not there yet. So I'm some place in between. So we physicists usually make a joke at this point that I'm in a superposition of two states and the wave function hadn't collapsed yet. But who knows if this joke will fly here. Okay, so yeah, and the one thing I want to ask you is to interrupt me at any time and ask questions. Let's not wait until the end if something doesn't make sense. This way, I will be able to better understand what is it that you don't know and need to clarifications. Okay, so this is a roughly outline of my talk, quick introduction, then quick theory, and then bulk of the talk will be mostly about phenomenology. And that is what that paper that I'll be talking about is about. I will mention at the end some experimental results, although in biology we should say observational results. Although you can think of experiments that one would do, and I'll talk about that as well. But the bulk of the talk will be about phenomenology, then I will quickly conclude. Now, so there is a huge literature on that, and not suggesting you go ahead and read every single paper that I listed here, but that may be a good outline for you to look at and see, right, what am I interested in? Am I interested to understand how the theory of learning works? Then you would go to this first paper. Are you interested to see why is the learning paradigm falls good and can describe potentially everything in the world? Then you may start here. Now, I don't expect most of you to be physics inclined people, but there are few papers in that direction as well. Many of the tools that I'll be using actually came from physics. And so if you see something, some term, something that doesn't quite ring the bell, I apologize for that, first of all. I think those tools and those words, everybody should know, regardless of where you got the degree in biology or machine learning. But if that didn't happen, I will try to clarify those terms. Okay, now coming to biology, which is the main idea of this talk. There are three papers here. One is theoretical paper. I call it the theoretical. Doesn't mean it's more complicated than phenomenological, but that is where the dynamical system that we are studying, biological dynamical system, would be more or less precisely defined. Now, as is, it will look complex. And so solving problems within that framework immediately without making approximation assumptions may look difficult. Nevertheless, that's a place to start if you again want to get a view of why do I think that everything in biology can be described through learning. Now, there is some numerical work that was done. It's actually very much on the intersection by bio-machine learning. You take a given biological phenomenon and see how it fits into machine learning algorithms. So it's right where you guys are. But most of the talk will be about this phenomenology. And once again, if you have questions, stop me at any time, interrupt. I'll try to clarify. Okay, so let's get started. Now, this is the picture that I think all scientists should have in their mind if they're trying to model anything at all. That is kind of the way at least physicists are think about modeling any given scientific phenomena. What I'm trying to do, I'm trying to advocate that this is way of thinking is useful, not only in physics, also in biology, machine learning, have some people iterating in psychology, all of those fields, all of the above. If you come to that framework where you separate the statements that you make within a theory, within an experiment, and within phenomenology, everything starts making more sense. And where do you start, right? So you have a given phenomenon and you want to model. The one way of starting is you actually look what is the mathematics or the fundamental thing that happens maybe on microscopic level that would lead to whatever phenomena I observe. And then if you do that, then you have to follow this top-down approach. Yes, you start with something very messy and microscopic, maybe easily defined, but messy in terms of solving it, but then you derive some simplified version of it that can be already matched to experiment, and that's top-down approach. Usually it doesn't, but you learn what things in your theory and modifying of which things in your theory, how it affects things in the phenomenology so that you can match your theory to the experiment. So you don't match directly theory to experiment, you match it through this phenomenological layer. And as I said, the bulk of the talk will be about that, about how you do it from top down, trying to understand what your phenomenology looks, and other way around. So you got a phenomenological theory, it doesn't match your experiment, what do you do? How do you adjust it at the phenomenological level by going up? So here is a learning theory of how to do science, and yet we are within it, we're going to be using the learning theory to actually model the phenomenon. So like some levels of complexity there. Now, if I haven't confused you enough yet, let me know, I'll try to work harder. Okay, so let's get going. Theory, as was promised, we'll try to go through that fast. However, that will build the foundation what we're going to discuss. Now, first, we are actually considering very general problem of learning, even maybe a more appropriate optimization, where I haven't told you what the mathematical framework is, whether it's going to be neural networks or maybe something else. But within that theory, we want to identify two types of variables. Any dynamical system has variables, and what we are doing at this stage, we're identifying two types of them. And the first type we'll call trainable variables, the ones that actually, whose dynamics determines on trying to optimize some function that we are going to discuss, and non-trainable variables. They just go there just because that's the way that this is the way the information is being transformed. Maybe they just take certain values, and the values they take is not because of they are learning anything those variables are trying to optimize, but it's because they describe maybe things you observe around us. To be more precise, and since we are kind of at least 50% of us maybe biologists, let's right away match it to something that we already know. Let's think that you are optimizing your function, trying to minimize it. It's very high and very complex, has lots of minima. And then you came to some point where it is always at minima, at local minima. And so the first derivatives of that function with respect to your trainable variables is small, but the second derivatives can be large or small. And in that case, what we're going to do, we're going to say, all right, so if the second derivative is very large with respect to a given variable, then the function, the learning dynamics is not likely to take you outside of it. So we are more or less going to stay in that value in the minimum of the function. So think about a parabola, which is very sharp, meaning second derivative very large. And we'll call those variables core. Okay, and the second type of variables is when the second derivative is actually not so large, it's smaller. And so we'll call those derivatives adaptable. Those variables adaptable means you can still go in those directions. For the core variables, you wouldn't be able to go anywhere. The adaptable, you can still go and explore. And then there are third type of variables when the derivative, second derivative actually is zero. And so, yeah, you can go in those directions, but that wouldn't change the value of whatever function you're trying to minimize. And we'll call them neutral variables. Now, in terms of biology, that may correspond to a type of variables like genotypic or phenotypic variables that you are dealing with. And then we'll discuss that shortly. We'll give you an example shortly. And that's Qs, QC, QA, QN. 3 types of variables. Very slow or almost frozen, changing a little bit faster and changing yet faster. And then you have non-trainable variables. They don't actually train, they just use to transform information, and we'll call them non-trainable variables. And for the non-trainable variables, they can be variables that describe environment around a given organism. That would be E superscript E, or they describe the state of the organism itself, then X superscript O. So again, 2 types of variables. This division turned out to be useful once we get to biology, and that's where we specify the division now. The fact that the variables divide into slowly and not so slowly changing variables is not unique to biology and can also be observed in machine learning systems. If you study how certain variables change, you will see certain jump wildly left and right without changing the loss function, and others are more or less stuck in their trained values. So I can talk more about the spectrum of those variables, but that would be going in the direction of more theory when I want to actually take us to phenomenology. Okay, so the other important point is that question whether something is fast or small only makes sense if you specify time scale. So if you take a time scale of a lifetime of an organism, that adaptable variables can be phenotypic traits that quickly react to environmental changes, so they change fast. And the core variables would be genomic sequences, so it's genotype variables that change minimally, if at all. And our neutral directions can be some directions that whose change doesn't affect the outcome. Okay, I spent a lot of time on this slide just because I want to set the stage. The last thing I will say, the abbreviation KNOE together stands for canoe. If you find it useful, that's fine. All right, now I'll try to go a little bit faster because I want to get to the phenomenology. And here is an example of how one would think about organism as an actual neural network. Now, this is a simplified toy version of what organism would be. This is the one that has only 3 layers. One corresponds to environmental degrees of freedom, standing right on top. Then you have phenotype degrees of freedom, and environmental degrees of freedom changing the fastest. Then you have phenotype degree of freedom, they change slower. Also neutral degrees of freedom, they do change, but they don't quite affect how you predict the environment. We'll get to that soon. And then there is genotype that are just frozen to certain values. They don't change at the time scales of the lifetime of organism, but they do change on the longer time scales. So now we want to take that and develop a theory, neural network, and see if the neural network theory is capable of modeling such systems. For that, we should first describe what the neural network theory is in as general terms as possible. So the idea is that we will treat, if we think about weight matrix and biases as trainable variables, and will be which correspond to Qs, and the state of neurons as non-trainable variables, then the non-trainable variables change according to equation 1. However, the trainable variables change according to equation 2. That is the main complexity which comes from with the theory of learning that is actually absent in the standard physics. In physics, we really only have one, where you have a state of the system and you know laws by which this state will change. It can be the Hamiltonian, a classical Hamiltonian system, or quantum Hamiltonian system. It's all the same, really. Now here, what we have a dynamics that actually allows a new type of dynamics, and that we'll call trainable. Something like that does happen in gravitational physics, although, of course, I won't have time to talk about that. All right, so now here I give you some examples of the loss function. Loss function can be at the boundary of the organism, and so it's only the environmental degrees of freedom and change from how organism predicts the environmental degrees of freedom affect the loss. Or in the bulk, inside of the organism, there can be, again, some degrees of freedom that by some reason the learning system decided to optimize. And so I will refer to those as kind of kinetic term, and this is a potential term, although again, this is a loose terminology. The point is there is some function of x and q's that describes loss function in machine learning, but how does it correspond to something that we are dealing with in biology? Okay, so take this bulk loss, which presumably has all possible terms. This potential can be very complicated. There's many x and q's. And the question is, what does that correspond to in the terminology and things that we do know and measure in biology? So the first thing is kinetic term, and you should think about this as ability of organism, I already said, to predict the state of the environment. So if the system is a good in predicting environment, presumably it can survive better, etc. Not direct connection yet, but yet this is why optimization of the ability of organisms to predict the environment is important. Now, the potential term may be just compatibility with the environment. Now, some organisms may prefer to live in one kind of environment as opposed to the other one, and that would generally be described by this potential term. Now, there is a term that is optimized in biology, fitness. Malthusian fitness is one example and expresses reproductivity or success of reproductivity of a given genotype. So that is the one we're going to match to loss function. Now, setting loss function just to be fitness turns out to be incorrect. It doesn't have the right properties. And so really what you want to set it to log, which is what you have here. Now, the other difference between loss function and the fitness that you really want to minimize the loss and maximize the fitness. And so that's where the minus sign comes from. But other than that, there's two functions, loss function and the logarithm of fitness, which is sometimes called additive fitness. There can be arbitrary parameter which multiplies the arbitrary constant of proportionality. I'll just put T here. Now, I put T and it does start looking like a temperature, not because I think I have shown that, but because of what I'll show ahead of us. So it turns out that there is a well-defined coefficient of proportionality between H and log F, and the coefficient is the temperature. Now, I'll just discuss two definitions of temperature, and for both of them, that is actually the right proportionality. And looking ahead, what really will happen is that the inverse T will become so-called Lagrange multiplier, something that you would use if you're trying to solve optimization problem. You're trying to extremize something subject to some constraints. And the method is used Lagrange multipliers, and that will play the role of that multiplier. Details are below. So if you haven't understood why T corresponds to temperature, you're absolutely right, because I've just showed you something that will happen below. Okay, so now neural network, and let's have again a cartoonistic but overview of the entire biosphere. So there are some connections between all of the organisms. There is environment. There are molecules that are interacting within environment. Molecules build up cells. Cells build up organisms. Organisms build up population of organisms. There is entire biosphere. And so all of those degrees of freedom interact with each other. And the main point here that those degrees of freedom still split on these two types, the ones that are being trained and the ones not being trained. And the idea that this being trained and not being trained actually happens on all levels, starting from molecules and below, if we're interested in physics, and going all the way to upper and upper scales. Now, this idea that it happens on all scales actually comes from physics. The idea, not that it happens on all scales, but that you can describe the system, the same phenomena, the learning phenomena, simultaneously in all scales. But the only difference is that some phenomenological coefficients would change, but the general description would be the same. It's called renormalization group in physics. So here we are applying this idea, but what we are saying that learning happens on all scales. Once again, I'll have a reference to actual machine learning experiments that you do, and you observe that the different variables change on different scales. And there are some of them that describe short distance, high frequency, small time evolution. and others describe low frequency, long time evolutions. So that happens in both in biology and something that we have observed in machine learning.
Sorry,
but then, yeah, there's a question. Go ahead.
See, okay. About renormalization, I'm not very.
You're not fluent in renormalization group flow. That is okay. I just flushed the word to say where the ideas come from. Yeah.
Okay, I'm only a bit lost about that word specifically.
Oh, yeah. No assumption is made that you know something about the renormalization group flow. Everything that I would want you to understand will be explained. The reason I say that is just tell you where the idea came that the learning doesn't happen only on one scale. Now, in biology, we have also there's some phenomena that people call as multi-level selection. So that is relevant to that. Now
Can I ask a question?
And of course, yeah.
So hi, I'm Sal. So I'm interested because you said learning happens on all levels, whether trained or untrained. What how do you account for that?
Okay, so I can answer any of those questions with putting my biology hat on, machine learning hat on, or physicist hat on. Which one do you choose?
Machine learning, NLP, more my speed, and I'd love to get your notes, the presentation, if you have a PDF that.
No, I don't have notes. It's all in the papers. But let me answer the question from the point of the machine learning, right?
Yeah.
So machine learning, you start your machine learning task. You wait long enough. Wait until your machine learning system came to what I call a learning equilibrium, where the loss function doesn't change much, but it's kind of fluctuates in the middle. And then you study the behavior of different variables. What you observe is that there are different variables, trainable variables. They change on different scales. Some change on short scales, small scales. They're fluctuating very rapidly. The others change only on the larger scales, that they are slowly changing, and so you have to wait long. And so that's multi-scaling. That means that the system is actually converged to a state where a range of scales is relevant. Now, if you have a shallow network, that may not happen. For deep, it will definitely happen. This is one way of understanding why the deep network would be good. Basically, trainable variables are trained to look for all scales, features on all scales, and that's kind of what makes it so. Yeah, so with my machine learning hat on, all I'm saying that there are not isolated subnetworks that we have in biology, although you can try to allocate that as well. But the variables, the trainable variables, they are always learning on some level, on some scale, with some frequency. And there is a wide range of scales for which they learn. Other questions? And I haven't started the talk yet, actually. Okay, so that's all right. That's good. All right. So anyways,
thank you.
Yeah. But again, I mean, I understand this is not conventional way of talking those things. So there is, again, I gave similar talk. It took me 3.5 hours to Freeston's group of neurobiologists. So here we have a mixture of machine learning and bio. But again, if you ask a question, tell me with which approach you want me to answer it, and I'll be more than happy to answer. Okay, so now this is a crucial point because I've started telling you about those slow variables, fast variables. What do you mean? Of course, it's with respect to some scale. So you said, I'm looking at a certain scale, and then once I look at that scale, some variables change fast, then the scale, some change slower than that scale, and others just change with the same frequency as the scale I've chosen. And so, but then the answer actually depends of what you call a scale. So am I interested in evolution of biological system at the in 1 second, or do I am interested in 1 hour, or in 1 day, or a lifetime of a person, of organism, or maybe thousands years, millions years? And for each of those scales, if you want to answer the question, then what we do in physics is a well-defined prescription. So you take your scale, You look at all scales that are higher than that. You assume that those scales come to an equilibrium. You integrate them out, throw them away, and then you get an effective description where those small high-frequency fluctuations are just present only at the phenomenological level of adjusting some parameters. This is called a renormalization procedure. I started with a macroscopic series that has all the scales. I was not interested in that scale. I was interested, let's say I'm interested in the time scale for my lifetime. Then I have to integrate out, average over all of this microscopic dynamics. And I can, keep asking the same question and integrating out more and more degrees of freedom, and then it will push me more and more to a larger and larger temporal scales. You can do it in physics. You can do it in biology. You can do it in machine learning. You have a machine learning system. It looks complicated. You don't know what's happening. You're trying to come up with a simpler version of that system, which you can handle and understand. You integrate out under assumption that something came to the equilibrium. Okay, so now we're actually going to do it. So now all this was preview to the actual stuff. Now we're actually going to do this phenomenology, because integrating out degrees of freedom, moving to some level, averaging out over some small scale, high frequency scales, is the phenomenological approach. And in physics, there are no alternatives. There's no hope for us to write it down actually how, if you've seen a standard model Lagrangian, it's huge. And like, if I am to pay attention to all of individual fields and particles, it's hopeless. I won't be able to accomplish anything. So what we do, we integrate out. We make an assumption about everything else, and then concentrate on something we're interested in, on one thing at a time, 2 things at a time, 3 things, as many as we can afford, but not on everything. On everything, it would be what I call theory. Once you've already integrated out some, I'll call it phenomenology. Okay. All right. So now, so let's now have a concrete description of what would be like a phenomenological approach to machine learning. So there are already, regardless of what scale you look at in machine learning, there are already these 2 types of variables. There is states of neurons that evolve through the activation dynamics, and there are trainable variables that evolve through training dynamics, 2 different types of dynamics. What you can do, you can say, all right, let's study a minute when the activation dynamics is fast and the learning dynamics is slow. Meaning you are just taking your mini batch size to be larger, right? And then you keep updating your your training data set, and activation dynamic goes through your neural network. States of neurons quickly change, and that's all fine. And then you go through the entire training data set, and everything changes. In biology, you go through the, I don't know, maybe 1 year of observing the environment, and it's again kind of a training data set for the biological organism that is learning. And you just average over this. You're saying, well, the best I can do is actually average out and try to come up with my best understanding of what the environment would be statistical, or what the training data set is statistical. But what is the right ensemble should I put for those things that I'm trying to integrate out? What is the right, like, let's say you are training data set with NIST data of handwritten images. And then there are many images that is in your training data set. There are other images that are not in your training data set. And you still want to assign probabilities for everything, for this whole thing. How do you do that? What do you do? So, and the proposal came from James, actually. He said, it doesn't matter what your problem is and what you're trying to solve. If your question is probabilistic and you're trying to assign probabilities to anything at all, first formulate what is it that you know about your system. Like, list down all of the things that you know and call them constraints. For example, I know my average loss is equal to such and such. I measured it. Okay, so you listed all of the things you know, you interpret them as your constraints. And now after that, what is the probability distribution? What he said, James, is that it has to be the maximum entropy distribution subject to those constraints. And that's called the maximum entropy principle. I don't think there's anything that can beat that. What it tells you briefly is that just do not make assumption about the distribution, something that you do not already know. And everything you do know, put in terms of constraints. And so this constraint is an example. You say, all right, so the only thing I know about my system is its loss function. Or for the biological system, the only thing I know is its average fitness, right? Fine, if you do that, then you go through 5 lines of solving optimization problem for using the method of Lagrange multipliers, very simple, and you derive equation 7. So if this is your constraint, then equation 7 describes what are the probability of different axes, different non-trainable variables, different states of neurons, different states of the environment for the biology, given some fixed set of trainable variables Qs. Okay, so that's maximum entropy distribution. You cannot beat that. Now, if you know something else, you should put it as well, and then there'll be more Lagrange multipliers, there'll be more terms in the exponent, but the idea will be the same. Now, here comes the physicist tool that, of course, invented by mathematicians, heavily used by physicists, and mathematicians even forgot that they invented it. So when they need to do it, they kind of don't use it as often as they should. It's called a partition function. We write down this integral that just looks like some mathematical thing where you integrate over x this distribution that you just derived. So you derive distribution, but then you integrate it. Now, roughly speaking, what 8 is a normalization constant. So if I divide this exponent by z, not roughly, it's exactly what it is, and then it would be legitimate probability distribution that it integrates to 1. But we don't care about that. We treat it as and we call it a partition function. It's kind of assign weights to all possible states of x. What's the probability of that? What the probability of that? Now, for example, if there is some state x for which h is very far away from u, then it will be very unlikely. If it's close to u, then it will be very likely. So if h is very, very large, then this is exponential factor that will be suppressed. So it's not likely to be in a state which has a very large loss if your loss is already small. So those fluctuations are kind of suppressed. Okay, what is it good for? Well, once you calculated the z as a function of beta, that's important, and as a function of q. So x gone because we integrated it out, but q and beta are there. Once you have that, from z, you can obtain all of the things that you can measure. For example, you can obtain average loss. So we usually take the z and define the quantity which we call free energy. But at the end of the day, once you have z, you have f. It is just plugging into logarithm multiplying by minus b inverse, beta inverse. But once you have that, you can check by differentiating this with respect to z. If you plug in z here, you will get average loss, meaning it will be an integral of h e to the minus beta h divided by the z, by the normalization. So it's an expectation value of the loss. You can also calculate entropy for it right away. Now it will also involve one derivative, but then you won't have beta here, you have beta squared outside. And many, many other things you can calculate, like what are fluctuations of loss, etc., etc., etc. Okay, so far, what I had in this first and a very important slide, I made a very important assumption that maximum entropy principle assumption. If you know something about system, write it down and use it as a constraint. If you don't know, assume that it is just satisfy the maximum entropy distribution. And after that, you can, for simple system, you can actually calculate this integral. Now, for a difficult system, you cannot calculate this integral, but you know what kind of terms would appear there. Okay, and so when we say theoretical calculation, we actually know exactly what this integral is. When I say phenomenological, we actually make assumptions about this integral, and we only calculate it in certain limits. Now, the truth is that in physics, we actually only know how to calculate one type of integral, and it's a Gaussian integral. Everything else is just becomes almost impossible to calculate, and we apply methods like perturbative method to all dualities. But for the Gaussians, when there are like x squared appearing there, that's easy to calculate. We do it right. Okay, so that's the setup. Now let's now move, and so this is top-down approach. We started with a theory. We managed to understand what kind of things happen in the theory. Now what we're going to apply now is actually bottom-up approach. It's purely phenomenological. What really matters is this u as a function of certain variables, as we will see. But to understand how that matters and how we actually can calculate this phenomenological function. By the way, this phenomenological function you can calculate for biological systems by observing your organisms long enough, for machine learning system by running your machine learning simulation long enough, and I'll show you how to do that. But the point is that once the system reaches the equilibrium, there is a first law of learning that needs to be satisfied. Basically, change in f must be set to 0, and f is the free energy. So you have to reach a point where this f defined here doesn't change anymore. It's df equals to 0. Now, this is actually the first law of learning. The second law of learning is a lot more interesting, and I haven't displayed there here, because it goes in a complete opposite from the second law of thermodynamics. So in thermodynamics, we know that the entropy usually grows. In a learning system, and we observe that, what happens is the opposite happens. The entropy tries to decrease. And there is a reason for that. Basically, you supply your network with the training data, and the network tries to learn it, right? Tries to correlate with the data. And then when you have degrees of freedom correlating, that goes into the decreasing of entropy. So to resum, just one thing if you want to remember about thermodynamics of learning is that there are, again, these two types of dynamics. One is the activation-like dynamics, and the entropy grows there. And then the other one is the learning type dynamics. That one is actually trying to decrease the entropy. And the two terms are fighting with each other. And so depending on your setup, your initial conditions, one or the other may win. Okay, so now let's think about what different terms here mean. So the term that appears here, T, is inverse beta. This is just your parameter beta that we just introduced in the previous. This is a Lagrange multiplier that we introduced on the previous page. Now, when you have T equals to beta to the minus 1, but you can also look at this equation. And if you df equal to 0, what it tells you that du ds is actually equal to T. And so that's what I write here. So let's say you're have some way of measuring the loss function of your machine learning system when you change entropy of the system. And you manage to discover how U changes with respect to S entropy. Then you can reconstruct function U of S. You can differentiate it. That will be your temperature. The same with biology. Remember, U is related to your additive fitness. Let's say you can measure the fitness of your system at different states. The same organism, but at different states. I'll discuss it later a little bit. Then again, you do the differentiation. You reconstructed how fitness changes when you change the environment. Yeah, because S is responsible for the environment, and then you will be able to reconstruct what is the temperature, biological temperature of such a system. Okay, so I'll get more to the phenomenology of this later. Let me just say that at this point, we integrated out all of the X's, all of the non-trainable degrees of freedom, because they're changing very fast. You can also try to get rid of the trainable degrees of freedom, all those Q's, and only treat them at the level of how many there are. Let's not be so microscopic. After all, this is our strategy. Let's just keep integrating more and more, and the more we integrate, the more in thermodynamic limit we move. So let's integrate, also get rid of all of the trainable variables, and only keep their numbers, how many there are. Then instead of what we call the free energy, there is actual grand potential that needs to be minimized. But really what happens is that you have this additional term mu dk instead of q dq, where q dq, we had the vector here. So there's a lot of trainable variables, where k, there's only one number, how many trainable variables there are. And again, there is some function U of S and K now. So you change the number of trainable variables, you change your entropy, and you say, okay, well, I know now how my loss function changes. or I change the number of trainable variables in my biological system. I change the entropy of the environment in which biological system lives. I reconstruct this U. There we go. I reconstruct this U. Now I can differentiate it and obtain both a biological temperature and now also call it evolutionary potential. Now, the larger the evolutionary potential, the harder it is to add new degrees of freedom. The smaller, the easier it is for you to add new degrees of freedom. So presumably a system for which organism which has a smaller evolutionary potential, it's easier for it to evolve. That's another thing that can be, again, checked against experiments or observations for certain data. Okay, so let me now actually dive all the way into the phenomenology. But before we do that, let me just match it to some other models that were already developed in biology. So for that, we'll start by considering an ensemble of organisms that all have different, they have different by the adaptable variables. So there's one type of the trainable variables differ for all of them, but the core variable or the constant variable is the same for all organisms. Basically what I'm saying, consider the same type of organism, right? And now we want to consider ensemble of those organisms, because now we again want to go to this phenomenological limit. Now in probability theory, they will tell you there are two ways to think about probability distribution. In the Bayesian, where you just assess your system, you look at it and say, all right, well, I think this coin is fair, probability 50-50. Or you do frequentist approach. You look at your coin, you throw it 100 times, it's landed 49 heads, 51 tails. You say, okay, well, you know, the probability is 49 versus 51. So this is your experimental data. However, if you take the number of trials to infinity, the two will agree. Now, in our case, when we are considering a population of organisms, we can never take the number of organisms to infinity. And so there is some number of organisms that currently is in within a population. In biology, I call it n sub e. All right, so we'll keep that notation. Now we'll make a full phenomenological model. We can make any assumptions we like, as long as their results agree with the experiment. The assumption we make here that we have a population of n e organisms, and they have k trainable variables. And the dependence of the number of organisms to the number of trainable variables is exponential. Just an assumption. So basically what it says is that if you have more variables, then you can afford to have a larger populations without having populations having identical states of those trainable variables and being basically useless for the evolution. So this is effectively how many do we need to actually train all of those k parameters. Just a phenomenological model. Again, we're going to talk about this. We see how it fits the data, and then we'll be happy. Now, at this point, now we only have a very coarse system, biological system. It has only two parameters, it has entropy and number of trainable variables. If you're talking about machine learning system, again, two parameters, entropy of your training data set and the number of, you know, weights, biases, whatever you want to call it. And now I say, all right, so it's hard for me to understand how the loss function will change with all of this, you know, its complexity, but maybe I will just look at it phenomenologically. I'll try to run my simulation many times and reconstruct and see what the loss function is as a function of S and K. You do that, you reconstruct the function, and here pops out right away what you will call temperature and what you will call potential, evolutionary potential. In biology, it would be called biological temperature and evolutionary potential. In machine learning, you can call this computational temperature and potential for trainable variables. You can also have a potential for non-trainable variables. Let's say you want the number of neurons to vary, and then that's a whole different talk. Okay. Yep, go ahead.
Hello. I do biophysics and stuff like this, but earlier you were saying that when two organisms share the exact same, in this case, genome or set of trainable variables, that it's useless, that it's not being very helpful.
Right.
But perhaps you're going to get to this, but in an actual evolutionary sense, having multiple redundancies means that when you have functions go through the population and purge them or remove useless organisms that no longer contribute to the overall progression towards whatever ideal state is required, having these redundancies helps for these bottlenecking events. How would your model?
Yeah,
Okay.
so I think what you're telling me is that this B is not quite of order 1. And I agree with that. So the B is, I mean, again, this is a really exponential model. So this is not like 1-to-1, and this is the beauty of phenomenological model. You don't describe things, on the microscopic scale. So you say, okay, well, there is B. We'll include it as a parameter and see how things work. Do they agree with data? Yeah. So phenomenology all the way, really. I mean, we understood principles of how theory gives us a statistical ensemble, and then it's phenomenology all the way. Okay, so now consider this, it's called Kimura model, but however, people mostly call it a Sella and Hirsch model, just to show what we are talking in terms of this biological system that people study. And we'll also identify where the mistakes those people made. Why did they call something temperature, which isn't a temperature? Because in statistical mechanics, there is no room for wiggle. It is very clear what temperature is. It's in a statistical description, it is Lagrange inverse of Lagrange multiplier. In thermodynamic description, it is a derivative of the average energy or loss function with respect to entropy. So there is no room to wiggle. So here's the model. Simple model is called ideal mutation model. We'll generalize it a little bit further, and then we'll discuss phase transition. Hopefully, I'll finish in 10 minutes, but hey, it took me longer than I thought. Sorry. So anyhow, so the step is this model, a more complex model of this type, and then phase transition, this punchline, how to just use this thermodynamic approach to describe phase transition, such as origin of life. Okay, so we have now N organisms, they're described by the genotypes that can undergo rare mutations on some time scale. Every time you talk about something, it's always the time scale, right? So they have a rare mutation, some fixed time scale we chose, and then followed by fast fixation, or much smaller time scale. So basically, organism after that all become of the same type, and then again mutation happens, and then fixation, mutation, fixation, and you have to wait for some long time for the mutation takes place, and the fixation happens very fast. That's Kimura's model, and here let's describe what it means in the terms of statistical ensembles. So you really have you want to write down the probability distribution of Qs. Now you really want to treat those Qs, although they are really trainable, but we are talking about them as not very much trainable. They are in equilibrium, the system equilibrate. So at those time scales, they're really changing fast. Those Qs keep jumping. We call them genotype, and then that's why I write Q, but really they are not all fixed and all they've established equilibrium. Then you just write down your partition function, just like what you had, this probability distribution that we had before, and you integrate out all x's. Once you integrate out all x's for each of and every one of those NE algorithms, you integrate all of them. And once you integrated all of them out, what you are left is with this function, where F here is the free energy. Remember, the free energy is minus 1 over beta log of the partition function. So this integral is actually a partition function. And so therefore, in terms of free energy, this is what it looks like. Probability distribution as a function of this FQ. And what you see in the exponent, you see two things that come one next to another, beta and NE. This is actually the assumption that algorithms do not collaborate with each other too much. There, it's called a decoupling limit in physics, where you can express Hamiltonian, which H plays the role of Hamiltonian, as a sum of terms of this xn's. They're not interacting with each other, only through Q. Anyhow, if you have that, then you have these two terms, beta and NE, together. NE is how many organisms there are in the population. Beta is the actual inverse temperature. Okay, so now I understand that you can confuse, because basically you can just call the whole thing T, 1 over T, or you can call just this T. Well, the Sella and Hirsch said that NE should be interpreted that as 1 over T, and that's incorrect, of course. This is only correct to the limit where we're not distinguishing of which goes to the left, which goes to the right of this product. Now, if we are to generalize the system and include interactions, the mistake that would make would be huge. So it's not the number of organism that is inverse temperature, but this parameter beta here should play the role of the temperature. Anyhow, if you continue, then you can rewrite now this distribution through the partition function of individual organisms multiplied by to the power NE, and that's what they saw, and that's why they kind of called this NE inverse temperature. Okay, now here we can also give a microscopic interpretation of what's happening. So this is Z of Q is a counterpart, macroscopic counterpart of the fitness. So that is the one that happens if you get rid of more parameters, if you integrated out more parameters. And the things that we integrate out, we just call x's. If you pay attention to those x's, then yeah, it is true that they will, then your partition function will be more fine-grained. You'll have a more fine-grained description of it. Now, if you don't pay to the details and integrate it out, then Z plays the role of the fitness. But here's an experiment that you can really do and see that this is correct. Now, a biological experiment, right? Actually, machine learning doesn't matter, the same thing. So you have two systems. You have the same organism. Yeah, let's talk about biology. We have the same collection of organisms, and you keep putting them in different environments. You put in one environment, they equilibrate. You measure it for a very long time. You put in another environment, they equilibrate, measure for a long time. And then within this equilibration, you see that the system goes through many fixations, from many states of the organism. Q keeps changing. Okay, and then for each Q, you measure its fitness. Okay, you measure a fitness Q in one environment and in the other environment. Then what it says that the ratio of logs so this is a fitness of organism given type and environment number one. You take a log of this. This is a fitness of organism Q in environment number two. You take a ratio of these logs. This is what it should be. Q's drop out and you just get the ratio of temperatures. So it means that it doesn't matter what I choose to be an organism, what fixed Q I'm looking at, this ratio should never depend on Q. And if that's the case, that's a confirmation of the result that beta is actually the right inverse temperature parameter, or T is equal to, and T is the right parameter for temperature, not N. And so potentially this can be done, but you have to run this experiment for a very long time and also be careful that the system is more or less closed. And of course, in biology, it's very hard. But this is at least as a Gedanken experiment can be done. Let's generalize it. And again, I'll go quickly now just to make sure that you capture the main point. So here was, for this model, the U of K is actually this. And so there is no explicit temperature dependence on the environment, no S. Now, here's a proposal of how, using some reasonable assumption, this is what proposal with this U should be as a function of both S and K. And then in addition to this exponent, you'll have also this. By the way, notice that this B gets rescaled by S in this particular phenomenological model. So there is an assumption that this B that we had in exponent before actually depends on the environment. It's not independent on the environment. Okay, so you take this assumption and then you do some important mathematical operation, Legendre transformation that, of course, no machine learning people, no biologists know about. But if you do that, then you obtain quantity, which is what could be grand potential, that can be used actually to check. So once you calculate this function, you can now check what should be the fluctuation in the number of organisms. What should be fluctuation in the fitness of organisms? How should it be distributed if I keep measuring a time sequence of the number of organisms, etc. No details are given here, but this is what you would do. So you come up with a model, and then you have a prediction of how NE would change. Not exactly how NE would change, right? But this statistical. Here would be the average value. Here would be this fluctuations, variance, third statistical moment, fourth statistical moment, as many as you like. Complete statistical understanding of how the number of organisms would change. And the same for the fitness, how the fitness would change. Okay, so we start phenomenology, we derive this expression, this is just mathematics, and once that we obtain something that can be matched to experiment right away. Okay, so I'm coming to an end, yeah, sorry taking me so long, but here is a punchline for the transition, and then one slide supporting the claim that the phase transition are happening all the time. Phase transition in this setup would be that you have some distribution before the phase transition, some ensemble, and some ensemble after. Okay, and then at the point of the phase transition, the two grand potentials that I've described here, those mathematical things, actually match. So what you can do, you can write it for the ensemble of molecules. This is well known, been tested. Now we've derived it also for ensemble of organisms. That's something we had in the previous slide. Now we can actually match them together and see what happens. When would it happen that ensemble of molecules all of a sudden will become ensemble of organisms? What will change? Those molecules will be larger, they will develop their own degrees of freedom, and all of a sudden at the phase transition, they will be starting sharing degrees of freedom. So there'll be, I learned some degree of freedom, I propagated to other molecules that no longer molecules, they actually develop this new scale. I am rushing because I'm Russian, but let me just tell you that one thing, just to show that this phase transition and development of new scale actually does happen using the data, the real data. So again, here we come to data. I'm a theorist, but I have to show you data. So what we took here, we took a coronavirus data in London at the early pandemic. We looked at all genotype of all the sequences that we could obtain in a given time, okay? And we measured distances, Hamming distances between them. So you take two coronaviruses, it's not likely that they will have the same, they will be the same, and so the Hamming distance will be 0. And so there's nothing here. Now, they're all coronaviruses, so the distance between them will not be too large, so nothing here. But there'll be a lot of coronaviruses that have distance, I don't know, it's 10 characters in your DNA sequences that have to be flipped to go from one to another. And there is some distribution. That's what you would expect. I understand very well this behavior. I understand very well that behavior. This one is because you have a high dimensional space. There's some notion of hyperbolicity comes in. This behavior is also can be very well explained because it has some equilibration. So the certain form of that tells you the system more or less equilibrated. But then, boom, something happens, and that thing develops 2 peaks, okay? And then it can develop more, 4 peaks, 5 peaks. Now, 2 peaks, which are temporary, they come and go. It's like new variants appearing. But they're constantly seeing a number of peaks that are kind of well separated is the appearance of those new scales that I'm talking about. Now, this is a later time phase transition than, of course, not the origin of life phase transition, but the idea is the same. Certain variables just make that transition of being independent to being collaborative. Okay, so I rushed a lot through the last few slides, although that kind of was the main point of seeing that you can really describe original of this phase transition. Let me quickly conclude, and then I'll go back to questions and can go back on the slides and screen. So there's a theory that I didn't have time to go over, but there's a separate paper on that describes, we go over all evolutionary phenomena and try to see how they can be explained using the methods, the terminology and methods of either both, actually, machine learning and physics. So there's like unification in a way of 3 things: a learning system, machine learning system, physical systems, and biology. So we went through all the biological phenomena to do that. The one interesting thing that we do see this learning appears on all levels. And so that's kind of was known, but I think now we understand it. Why is it beneficial for the learning system to establish that? And something like generalized central log model was shown, then again, I didn't discuss it. We were mostly concentrated here. So we showed what are the biological counterparts of temperature and chemical potential. I emphasize that this grand potential or the loss function as a function of its variables or its Legendre transform, which is called grand potential, both are equally important and useful. But that would only describe equilibrium. If you want to study non-equilibrium transition from one to another, then again, you can do that. And I've showed you the first phase transition, origin of life, as well as the other transition that may be occurring. Now, for the experiments, we have only seen this appearance of the new levels in the coronavirus. Lots more needs to be done. This is poorly understood, even at the coronavirus data where we have a lot, and we are investigating it now. Now, of course, we need more experimental observation, numerical tests of that, and there's some work that are being done right now as well. Now I'll just show you those three things that just one might think about. So it's really, I'm advocating for the statistical biology. And that is really phenomenology on its own is something that doesn't exist in biology and should, as well as machine learning, actually. So I'm advocating for the statistical approach to machine learning. It's much more complex than in physics because you have these two types of dynamics. I'm proposing that they should also look into artificial biology because if we are trying to identify some biological phenomena, why not study it at the level of the learning dynamics, like, maybe even at the level of doing machine learning tasks where you will see appearance of certain phenomena. Like, would it be beneficial for the machine learning system to have such and such algorithm that will learn from nature or not? Like, one of the papers we wrote is exactly that. We saw, like, is it beneficial for the machine learning system to be able to remove and add neurons? Yeah, we have that phenomenon in biology, program death, but is it beneficial for machine learning system? Collider biology is another one. It's, again, doesn't exist. If you have an equilibrium of one system and another one, and you're trying to collide to see what happens, that's, again, a lot to be done. All right, thank you. I'll just leave a link here in case somebody's interested in internship in the company that I've just started. But now I'll take all the questions you have.
Only one question. How do you calculate biological temperature on actual living systems, on actual populations? How do you do it?
Yeah. So the question is about calculating this. So you, for actual learning system, you are trying to calculate, let's say, the loss function, right? So then you want to calculate the actual fitness. How fit is a given Q, right? So you want to perform runs and do that, and then you can make this comparison. But I'm proposing something that can be even done easier than that, without actually going and identifying a system and looking at it. Just do what I said here. Put your system in a box as small as possible. So there are this called, what do you call this? The glass sphere where you put things and they live there forever. Make it as small as possible, and then monitor how the number of organisms of a given type change as a function of time. How it's done, it's another question. But in principle, you should monitor how this population change, and is a function of t. If you keep monitoring that from the spectrum of those fluctuations, you can reconstruct that function as well as u as a function of k at least. Okay.
Okay, thank you.
I have a question as well.
Go ahead.
So you brought up prior the assumption that everything's decoupled, all the organisms are not interacting with one another, but
and that was wrong assumption. Yes.
Yes, it's wrong assumption. And the thing the idea of that and the phase separations of itself kind of brought me back to the work that I do, because I work with intrinsically disordered proteins that undergo phase transitions, resulting in higher order assemblies spontaneously forming under various conditions, and how I control that and the like. And I guess I was wondering, if I was working with a group of organisms that, for example, something like ants or something, and they all had a genome that was fairly standard at the start, and they start mutating, but then they start forming additive interactions with one another. For example,
that's phase
that's the phase transition, exactly. And I was just wondering, how would you interpret structures appearing inside of these organisms? Like what? I meant less like separate, but more like co-additive structures.
No. I mean, that's the whole point. That's what we will see. So now, after the phase transition, you'll take all of your poor ants, you will reconstruct their DNA sequences, right? You will plot all possible Hamming distances between every pair. You will have some distributions, and you will see peaks. You will see peaks about things that are more collaborative. And so basically, the way you think about this is there a collaboration happening on all scales.
Yeah.
Okay? And then you have a small scale collaboration, like social system, right? Within a family, and the collaboration within your neighborhood, then friend circle, then collaboration within, I don't know, maybe people who share the same values, and et cetera, et cetera. So the same happens here as well. You have collaboration, it's a hierarchy, basically.
Yeah.
Right. The point is that it's not just we usually think it's like really discrete. It's, you know, humans all collaborate and there's just one level for all humans, but that's not the way it is. It's actually there's multiple levels, and then every such appearance of every such peak is expected to go through the phase transition, and moreover, it's beneficial learning. It means that the system decreased its entropy and temperature, and it learned how to predict environment better. So I think when you, with your ants example, you will just see this is the simplest way to do that. Now, another way of doing this is actually analyzing so this guy. So reconstructing that is another thing by observing how the number of them change before and after. You know, there'll be a sudden change in how this function behaves, how the number of in the colony fluctuates. So yeah, so there. That's it. Okay. Other questions? Oh, there's something in the chat. I don't know if there are questions, but no. Yeah, PDF from the talk. Yeah, I can share that. Sure.
Yeah, I was about to relay another message from the Discord server as well, asking for the same. If you could share.
Well, the talk ended here. Other stuff, it was just in case somebody asked questions. But okay, I'll share the whole thing. So let me. So should I just share in the chat? Is that?
Yeah, I can post it on the server and people can find it there. Thank you very much.
In the chat, or should I send it to you?
No, if you want to send it in the Zoom chat, I can repost it over in the community.
Sure. All right, let me do it. Let me do it now so that I don't forget.
Thank you very much.
Yeah. Any other questions? I will be more than happy to answer.
One more question. You said that it needs to be isolated, but in reality,
Never is. Yeah.
it never is. But in a simulation, it can be made so.
Yeah, simulation is not as interesting in the real world. Yeah, I think I can answer your question. If I can share, I don't think I can share that talk.
But
I don't think I'm permitted to send. So, but that's a good question. I'll just send you. Okay, so let me answer your question because I think it's a good one. Yeah, so you're never dealing with a perfect isolated system in biology. You're never dealing with a perfectly isolated system in physics, and also in machine learning. You're also very hard to isolate because of well, unless it is some simple data set where there's nothing that doesn't change. You know, Tesla car drives down the road, there is always noise. There is always something new you see in the incoming data. So isolation is never possible. We do the best we can. Okay, best we can to model this absence of isolation. Microscopically, it's very hard to model, and that is kind of has to do with the fact that this partition function integral becomes untractable, very difficult. But phenomenologically, it is okay. Even if your system is still open and is still interacting, it's still going to come to some kind of equilibrium. Okay, meaning that probability distributions will stop changing significantly on a certain time scales. Now, on large time scale,
No, my question was there could be a theory that could function by interacting systems.
Oh yeah. Okay, that was my collision biology. Okay, so now I'm actually answering in the order. Okay, so that is collision biology. I call it collision biology. Nobody calls it collision biology. So what I ascribe to you right now, two phenomena. One phenomena is when things equilibrate or close to the equilibrium. The other phenomena when this equilibrium system by itself through learning goes through the phase transition. Now, what I call collision biology is you have one equilibrium system and the other equilibrium system. And then you put them together, okay? Or three. And then you try to study how does that one affect this one. This is exactly what we do in physics as well. We have one particle that is kind of stays in its state, the other one stays in the state, we collide them and see what happens. And the success of this methodology only works if you have as much as possible control on the of equilibrating thing before this system and seeing equilibrating after. Now, however, once you are able to do that, then you can mix them up and study already non-equilibrium processes where there's some local equilibrium established here and here, but they kind of start together. I'll give you one example, which you know very well about, where exactly like this happens. It's called hydrodynamics. So in hydrodynamics, you never have a global equilibrium. It is only locally that you have a local equilibrium. You have some temperature well-defined and some a handful of macroscopic parameters that describe it. And then you move to a nearby point, there is another equilibrium, and there's a third equilibrium. And then you have all of them locally in equilibrium, but yet on the larger time scale, they start interacting. And that is the type of things that is modeled by physicists through the Navier-Stokes equation, hydrodynamics. So something like this can be happening in biology as well. You just assume that a local equilibrium for some subsystem, and then they start colliding and become, which drives away with them from a global equilibrium, but locally they're still equilibrating. Why is it a good assumption to make local equilibrium? The locally, you have fewer degrees of freedom, and for the fewer degrees of freedom, it takes less time to equilibrate. That's why this assumption works well in hydrodynamics, and that is why whenever I say, let's study small biological system, that is the reason. Because the smaller it is, the faster it will come to the equilibrium, the easier it will be for us to analyze what it behaves if it was in equilibrium, and then study deviations from it.
Thank you very much.
Sure. Okay, folks. I think I'll try my last time to actually share the slides, but if I cannot, I'll just send it. I don't know why it's not working. There must be some reason for that. Yeah, I think I'm just not allowed to share. Could it be? Yeah, that's okay. I'll just send it to you. Okay, this is it. Nicola, I think am I free to go? Or other questions, or what's?
Can you hear me, Vitaly?
Yes,
I'm so sorry. If I've been unresponsive, but my Zoom keeps crashing or not responding, and so I can't click things. I've been unable to click unmute
no problem.
until now.
I'll just send you the files, and then you can share it.
Thank you very much. I will stop the recording now.
Perfect. Thank you.